Good morning and welcome to the student poster session. I'm Rob Chavez and I'm here to introduce 4 student posters that we have today, 3 of which are winners of scholarships. I'm very excited. We're starting a little bit early, 5 minutes early from what was posted on the program, so we can give these students their time. They only get their lightning talks. They're going to spend about 5 minutes just briefly summarizing the posters. I hope you all took your time to read them. They're excellent posters. So let's get started. We're going to start with Katherine Swensen. She's our PhD candidate scholarship winner for the Nancy Weller Memorial Fund. And so take it away, Katie.
Great. Thank you so much. Morning, everyone. My name is Katie Swensen, and I am a PhD candidate at Washington State University. And today I'll be presenting my preliminary findings for my research that is supported by the Nancy Weller Memorial Scholarship. So as you can see from my title, Impacts of Wildfire Reburn Events on Water Storage and Transport in the Washington Cascades, my research focuses on the headwaters hydrologic dynamics rather than the surface water bodies. So I'll give you a quick nickel tour of my poster. So if I could get the next slide, please. This will be a zoom in, the background section. So the motivation for this research is derived in the climate-induced stresses on our hydrologic systems in the West. Not only are snowmelt snowmelt rates accelerating in the spring, snowpack is also declining, and then we are also experiencing an increase in wildfire activity in some places in the West. And this increase in wildfire activity impacts down gradient ecosystems by sedimentation and erosion, and it can also disrupt the timing and magnitude of water availability to down gradient streams and lakes. And in Washington State in particular, wildfire activity is projected to continue increasing through the end of the 21st century. And we can see that in the upper right figure there, with area burned on the y-axis and then time on the x-axis from the 20th century in that blue box through the end of the 21st century in the red and orange boxes. And this increase in wildfire activity in Washington has led to reburn events in some ecosystems that are historically adapted to much longer wildfire return intervals. And so an example here is shown in the bottom right figure. And you can see that there are 3 severe wildfire burn perimeters that are overlapping. And this is on the south slope of Mount Adams, which is my field site. And these wildfires occurred between 2004 and 2015, with the previous wildfire not occurring for more than 200 years in the past. So these events are unprecedented, and we don't really know how these reburn events impact hydrologic recovery in these systems. And so that's the aim of this research, is to quantify that impact. Next slide, please. So my field sites are on Mount Adams, and I have a series of plots that were either unburned for more than 200 years or burned once, twice, or one site that was burned 3 times. And so I draw your attention to the far right 3 pictures. Those are the sites I'm focusing on in this poster, the single, double, and triple, most recent burn in 2015. At each of these sites, I am monitoring snow accumulation and ablation using snow depth poles shown in that picture on the right. That orange pole in the far background is my monitoring pole, and then I have game cameras mounted on trees. I'm also monitoring soil, vegetation, meteorological data, and hydrologic data in order to quantify the seasonal water balance in each of these sites. Next slide, please. So these are the very preliminary findings of my research, and I wanted to point out 2 main results that I thought were particularly interesting. So the first is in the top half of this slide. On that plot on the upper left, we have major snow events occurring over the snow season. Those lines are representing the single burn site in the yellow, double in orange, and triple burn in the red. And we can see that first point is the first initial snow cover. And so all the sites have snow cover at the same time. Next data point shows the last full day of snow cover. And then the third set of points shows when snowmelt is complete in the spring. And so the takeaway here is that spring snowmelt occurs almost 2 weeks earlier in the triple burn site than it does in the single burn site. But the double burn site melts almost a week after the single burn site. And this could be because of a bunch of downed coarse woody debris that you might be able to see in the far right picture of the double burn site. And then the second major result I'd like to draw your attention to is shown in the bottom right figure. So we have the summer hillslope water partitioning in the single, double, and triple burn sites. On the y-axis is the percent contribution of the cumulative water balance, cumulative water year precipitation. And so the blue represents potential evapotranspiration, the red is soil storage, and the yellow is the residual. And the residual could be evapotranspiration that's occurring outside of the snow-free period, but it also is the runoff and the deep drainage that's occurring from these sites. And so we can see in the double burn site that there's a higher component of that precipitation that is in the residual. But we only see evidence of runoff in the triple burn site. So that image is some rills that are present in the triple burn site. So my hypothesis is that the double burn site actually has a lot more deep drainage. And then next slide, please. And so we see evidence of early melt in the triple burn site. We see evidence of runoff in the triple burn site. And then we see evidence of these large outflows in the double burn site. And all of these have implications for early season flooding in downstream or down gradient ecosystems, and then lower late season flows in watersheds that have experienced more frequent wildfire. One potential benefit is those coarse woody debris, the downed logs in the double burn site, which may partially counteract the impact of that earlier snow melt that occurs in our reburn sites. And so the next steps of this research are to incorporate a physical hydrologic model to better partition those outflows into runoff and deep drainage. And then I will be incorporating some climate projections in order to look at the hydrologic fluxes under a future climate. Thank you.
Thank you, Katie. That was excellent. We're going to move quickly to our next presenter, Salvador Rob Chavez. Salvador is our MS Student Scholarship winner of the Dave Lamb Memorial Fund. And so take it away, Salvador.
Thank you, Rob. Hello, my name is Salvador Rob Chavez, and I am currently a graduate student at Washington State University, Vancouver, pursuing an MS degree in environmental science. Today, I would like to share with you the project comprising my master's thesis work entitled Broadscale Distribution, Abundance, and Habitat Association of the Asian Clam, Corbicula Fluminea, in the Lower Columbia River, USA. Here, I will briefly summarize background information, my methods, and my preliminary results regarding the distribution and abundance of C. fluminea throughout the Lower Columbia River. But first, I would like to acknowledge my co-authors and my advisors, Dr. Steve Bollens, Dr. Gretchen Rollwagen-Bollens, our always helpful lab manager, Julie Zimmerman, of the WSU Vancouver Aquatic Ecology Group, as well as Tim Counihan of USGS for all of their guidance and support in this project. So a bit of background, Corbicula fluminea, commonly known as the Asian clam, is an invasive freshwater bivalve originating in Eastern and Southern Asia, which has successfully established itself in many lentic and lotic ecosystems of temperate and tropical regions around the world. C. fluminea establishes itself quickly in novel ecosystems due to its hermaphroditic reproduction and is thought to have deleterious effects on invaded ecosystems. as well as human infrastructure and aquatic systems. These effects might include the alteration of sediment dynamics through bioturbation from their pedal feeding activity. It could also include outcompeting native bivalve species in our area. An example that comes to mind is unionids for food and space, and clogging up human infrastructure like heat exchangers or pipes with their mucoid secretions and larvae. The invaded range of C. fluminea includes the Columbia River and its impounded lacustrine areas, and it's been known to be present there since about 1938 due to human transmission vectors like shipping ballast. And has since become well established. Previous studies have recorded main channel densities up to 722 individuals per square meter and nearshore densities up to 500 individuals per square meter. But despite these negative effects on invaded ecosystems and 80 years of occurrence in the Columbia River, the literature regarding C. fluminea in the Columbia River is extremely limited. Thus, our ongoing study seeks to expand the existing body of research by addressing the following research questions. Next slide, please. So first question, what is the broad scale distribution and abundance of C. fluminea in the lower Columbia River? The second question being, which environmental variables are the best predictors of C. fluminea abundance? And the third, how does C. fluminea morphological condition vary spatially in relation to these environmental variables? Next slide, please. So our study area. From 2017 through 2020, we collected adult and juvenile C. fluminea from 26 nearshore, shown in the top panel there, A, and 15 main channel sampling sites spanning 537 river kilometers of the lower Columbia. At each site, I also measured a number of environmental variables, including dissolved oxygen, pH, water temperature, salinity, specific conductivity, water depth, geographic location, chlorophyll A concentration, riverbed slope, and sediment samples to determine size fractionation and organic matter content. At the shore-based sampling sites shown in Figure 2A, triplicate 1 meter square samples were taken at 5 meter intervals at a water depth of 30 centimeters consistently, with all C. fluminea individuals of shell length greater than 2 millimeters collected from the top 15 centimeters of substrate. And then at the shipboard sampling stations within Bonneville Reservoir, shown in Figure 2B, that sampling was carried out on board the USGS RV Merlin, utilizing triplicate Ponar grab samples deployed from the deck down to water depths of up to 30 meters. And on the next slide, we'll take a look at some preliminary results regarding the nearshore distribution and abundance of C. fluminea as sampled in 2019. Next slide, please. Oh, you got it already. All right. The 78 observations of C. fluminea abundance from 2019 shore-based sampling ranged from 0 to 427 individuals per square meter. Figure 4 here displays the abundance of C. fluminea as nested circles, and these represent triplicate samples taken at each sampling site throughout the Columbia River. And the pattern of distribution shows a majority of samples with abundances greater than 100 individuals per square meter were located west of Bonneville Dam and near the most developed areas of the river, while the majority of samples with 0 C. fluminea abundance were located to its east. The station featuring the greatest average abundance of C. fluminea was near the Sandy River confluence at Gresham, Oregon, and that had 342 individuals per square meter averaged between the 3 triplicate samples. And these observations of abundance represent the broadest spatial scale of C. fluminea sampling to date in the Columbia River and are generally consistent with, albeit slightly lower than, those made in the 2 previous studies known to us, both of which were more limited in the spatial extent of their sampling. Next slide, please. With 2020 shore-based sampling effort ongoing, future work includes sample processing to characterize sediments qualitatively by soil type and quantitatively by size fractionation and total organic carbon content. This information will be included in mixed model regression analyses to explore the relationships between habitat characteristics or environmental variables as predictor variables and C. fluminea abundance and morphological condition determined via their link to mass ratio as dependent variables. By elucidating relationships between C. fluminea population biology and habitat characteristics in the lower Columbia River, we aim to provide natural resource and infrastructure managers with valuable information on where, when, and why C. fluminea invades temperate river ecosystems, and we expect our findings to be relevant to other invasive bivalves, such as dry-setted mussels, which utilize similar means of reproduction and dispersal. So that concludes the summary. I would like to thank WALPA for its support of this research through the Dave Lamb Memorial Scholarship, and USGS staff for their logistical and technical support of this project. Thank you.
Well, thank you. Am I on? Jesus. I am. Thanks. All right. Next up, we've got Krystal. Krystal Saunoa is ready to give her presentation, so take it away, Krystal.
Yes. Good morning. Thank you. My name is Krystal Saunoa. I am an environmental science student. Can you hear me?
Yeah.
Yeah. Okay. I am an environmental science student at University of Idaho. I'm in my undergraduate. The project that I worked on this summer with INBRI allowed me to work with Dr. Frank Wilhelm from University of Idaho College of Natural Resources. And we were looking at the effects of boat wakes on the nearshore of waterways, specifically the Spokane River in North Idaho. And I will go over a little bit of our project during this presentation. Next slide, please. For the background, eutrophication is a very complex global environmental issue, and we are studying cultural eutrophication, which is the increase and increase of different nutrients into the waterways that are caused and attributed mostly from human activity. So water sports over the past 80 years has become a very popular recreational activity. And since about the 1980s, boat technology has been improving to where boats can now make intentional waves for different recreational sports. So we are studying the Spokane River, which is a very popular site in North Idaho, and we are studying the connection near the Spokane River and the Lake Coeur d'Alene. So our focus area in this study began with turbidity, which we are going further into this study with different phosphorus levels and trying to create a turbidity-phosphorus relationship. But up until this point, the scope of the study has solely been focused on turbidity. So phosphorus is typically a limiting nutrient in many freshwater systems, and with nutrient resuspension, which is related to turbidity, there can be an overgrowth of cyanobacteria because of the phosphorus release from the sediment resuspension. Next slide, please. So we wanted to choose an area on the Spokane River that was kind of representative of a natural riparian zone. And we were able to find that. And again, we're located, if you check out the map on the very top under site selection, we're located right at the mouth of the Spokane River that leads from Lake Coeur d'Alene. We're directly south of the Blackwell Island boat launch. And we are outside of a no wake zone, so that does give us the chance to experience the actual boat wake versus wave activity. With our site selection, we wanted to choose a site that was more representative of the riparian zones that did not have any additional armoring or any sand addition that was from humans. So we wanted to have something that had that representation to where we can see the natural sediments of the area, and the site that we chose did have a sediment characterization of mostly silt and a little bit of sand. And when we did set up our project, we had to make sure that we were very consistent with our data collection, and this project so far was, over the summer, was mostly focused on data collection of turbidity. So we used a few different ways to collect data that we were focused on. We used a turbidimeter to measure turbidity, and then we also used a water level logger. And we wanted to measure the different boat wake activity compared to the natural wave activity. And with our data collection, our sampling method, we took water samples at 15-minute intervals, and we measured the turbidity. And then we also counted every boat and documented the type of boat and the time the boat past our location. That way we can kind of match the boat activity and the frequency of boats and the disturbance of the wakes and match it with our hydrographs from our water level logger. Next slide, please. Okay, so our results, very preliminary, but our results did show that there is a correlation between boat activity and wave versus wake height. So the very top graph is a hydrograph from August 7th through August 8th. And this took place over midday, about 11 a.m. On August 7th, overnight into the morning of August 8th. And we can see that there is substantial wave and wake activity during the daytime. And it does taper off, and there's hardly any wave activity overnight. And then again, it picks up in the morning. And the very first, I guess you would say, the first activity that you see on August 8th, around 6 a.m., that is fishing boat activity. And below we have a chart that we were tracking the type of boats and the time the boats passed. And we could see that just from August 7th, from 3 p.m. to 8 p.m., we had a total of 225 watercraft that did pass our area. And that is what is representative in the hydrograph above, with all of that wake activity. I only did track the watercraft. I didn't track kayaks or like smaller personal unmotorized watercraft. But from going here on out, I think we are looking into tracking that type of activity just to see the type of use that this area is getting on a daily basis in the summer months. Next slide, please. So here we have a graph that shows the average daytime turbidity. And we start out early in the morning, and we can see that turbidity is less than 10 NTU. And that is representative of what the background turbidity normally is. And then we see a steady increase throughout the day up until about 9 from our last measurements. And then we can correlate the turbidity increase with the boat counts and the traffic and the boat disturbances that take place. With the graph off to the right, we see that correlation with increased turbidity and increased boating activity. And this is a very typical representation of a summer day throughout the busy summer months, where the background turbidity is standardly below 10 NTU, but then it does increase over the daytime. And most of the days that we did study included weekdays. So this is a phenomenon that takes place just daily in the busy summer months. Next slide, please. So our conclusions up until this point, we do see that there is a correlation between boating activity, wave and wake height, and boat wake height, the wave frequency, and the magnitude of the disturbance from each wake, as well as the increase of turbidity. So that's correlated together. So we do want to study further into this project, and we want to get a total phosphorus analysis from all of our water samples. And the point of this project is to create a turbidity phosphorus relationship to where we can be able to predict the total amount of nutrient of sediment resuspension and nutrient reintroduction based off of just a few samples of the turbidity in the set area, and then also the sediment analysis. So depending on what type of substrate is located in an area, whether or not it's a silt or a clay or sand versus gravel and more sandy areas, we want to be able to create a relationship to where we can use this relationship as a tool to predict the type of nutrient reintroduction and sediment resuspension in waterways. I would like to acknowledge Idaho Inbreed for funding this project. And I also would like to acknowledge Professor Vanmiddlesworth, who is at North Idaho College, who did help with a lot of this, the data collection of this project. Thank you.
Thank you, Krystal. Next up, we've got Hannah Hockenstad. She's our undergrad scholarship winner. And let's go, Hannah.
Awesome. Thanks, Rob. All right. So I'm Hannah Hockenstad, and I am an undergraduate student in the environmental science program at the University of Idaho. And Frank Wilhelm is my co-author. And today is going to be I'm going to be showing some of my preliminary results for my senior project right now. So my project was on the spatial distribution of zooplankton in Willow Creek Reservoir in relation to hypolimnetic anoxia and the implications for pelagic grazing. All right, next slide. So zooplankton densities and population fluctuate in response to lake chemistry changes like hypolimnetic anoxia. And one of the species that's pretty impacted by that is Daphnia. And that's because they go through diurnal vertical migration, and that can be interrupted when the hypolimnean becomes anoxic. So that would force them out of their deep and cold water refuge, out into the water column, and potentially out of the pelagic. So what we're looking at here is really, what do Daphnia do in response to hypolimnetic anoxia, and how does this change their grazing pressure? So my study area was Willow Creek Reservoir in Heppner, Oregon, and it's in like Eastern Oregon. And then next slide. And my objectives were to examine the relationship between hypolimnetic anoxia and the spatial distribution of Daphnia. Where do they go, and what do they do when hypolimnetic anoxia sets in? And my other objective was to measure Daphnia size and track those changes throughout the summer. So I was testing the null hypothesis that the vertical and horizontal migration or distribution of Daphnia is not related to the change in dissolved oxygen concentrations with depth in Willow Creek Reservoir. And my prediction is, as those deep water refuges become anoxic, they're no longer inhabitable for zooplankton. So they're gonna boogie on up the water column and potentially migrate out towards the nearshore areas to evade predation from fish. Next slide. My methods to collecting my samples were to collect vertical and horizontal samples of zooplankton in triplicates along 2 transects from the pelagic to the nearshore, and then collect physicochemical data. And that was temperature and dissolved oxygen. And then so in this figure to the right, that's where my transects are, and those are my sample sites. In this presentation, I'm going to talk about the data that comes from the 2 that are highlighted in red, and that's MS1, which is my pelagic site, and MS3, which is my nearshore site. Once I got my samples, I counted the zooplankton, I photographed them, and then I measured their body size in ImageJ. Next. And then here are my preliminary results. We're going to start from the top of these and move down, and we're going to start at the left one. Those top 2 graphs are the profile data. And so the blue line represents temperature, and then the red line represents anoxia. So on June 9th, anoxia hasn't hit yet, and then Daphnia are in it's the turquoise color down in the bar graphs. And so the density of Daphnia at site MS1, my pelagic site, are approximately equal to the density of Daphnia at MS3. So there is no significant difference between the Daphnia densities of those sites. But when we go over to August, that's when hypolimnetic anoxia has hit. And you can see that through that red gradient graph at the top. And then when you look at the bar graphs, you can see that the density of Daphnia in MS1 is significantly less than the density of Daphnia in the MS3 site, which is the nearshore site. So there is a significant difference between the densities of Daphnia between the pelagic and the nearshore. And then when we look over at this next graph, this is the distribution of the cohort sizes of my Daphnia. I broke my Daphnia sizes up into cohorts and then graphed them like this. And you can see that there's really no significant changes in their body size depending on their location. It doesn't matter if they're in the nearshore or in the pelagic, they're approximately the same size. And then next slide. And then this is the p-values for the t-tests for the changes in density and then size between those pelagic and nearshore sites for June and then August. So like I said, in June, there is no significant difference between the density or size with an alpha of 1. And then when you look at August, there is significant difference for density of zooplankton in density, but not in size. Then next. So upon initial analysis, we can reject the null hypothesis. There is a significant difference between the densities of zooplankton between MS1, which is the pelagic, and MS3, which is the nearshore. There are no significant differences, however, with an alpha of 1 in Daphnia size between the pelagic and the nearshore. And then going forward, I'm going to be counting the final set of samples that I have from 27th of September. Those will be counted, measured, and analyzed with a t-test, just as the rest of this data was. And then I'm going to go and do an ANOVA test to see if there is a significant difference between sites and dates. And then I'm also going to calculate grazing pressure for each site. And that data will really inform the difference in grazing pressures between the nearshore and the pelagic sites and the implications for the growth of algae when those pelagic grazers are absent, since they are more in the nearshore areas. So our results to date strongly suggest that pelagic grazing pressure is reduced during anoxia. Next. I want to thank the U.S. Army Corps of Engineers for field and logistical support. And I want to give you a big thanks, WALPA, for considering this project and awarding it the undergraduate research scholarship. It truly made my year. So if you have any questions, feel free to contact me at this email. And thanks so much for watching, tuning in.
Well, thank you so much, Hannah. That was really exciting for all four of you. So we don't have a lot of time, unfortunately. I wish I would have scheduled more. My bad. But here, a quick one. Katie, you ready? I'm going to put all three questions in one. Did you evaluate the time between burns, the severity of the burn, and the soil and/or the soil organic matter and its effects?
Yeah, so I didn't, but a previous student on this project did. So the time between fires, well, I suppose we didn't analyze that quite as much, but we have the single and the double that were burned most recently in 2008. So we can compare more recent time after fire and then longer time after fire. And then severity was, it was severe at all of the sites. And then soil organic matter was statistically significantly higher in the unburned site only. And then all of the others were statistically similar.
Well, great. You answered it perfectly. Thank you. Salvador, you're up. Did you evaluate river flow or macrophyte presence in Corbicula distribution?
Thus far in the study, we have not incorporated river flow or macrophytes. Macrophytes is something I did not consider, but river flow is something that I have been. If I can get that GIS data, you can bet that I'm going to be incorporating into the study. So thank you for the suggestion.
Well, that sounds like a good plan. Okay, Krystal, will you evaluate the effects of shoreline hardening and the difference between wake boats and other boats?
I'm not familiar with the shoreline hardening. I don't know what that means.
Oh, that would be bulkheads versus natural shoreline versus, you know, the bulkhead. Yeah.
Oh, yes. I mean, that is a great thing to actually observe. So maybe yes, I will talk to Dr. Frank Wilhelm to ask him.
Good. All right.
And then the other part of that question.
The other part is about wake boats and how those waves are bigger, and have you separated out sort of the differences between motorboats?
Yes, that's actually what we were doing. So we were focusing on boats that would create larger wakes and larger disturbance. And we are matching those boat types with the hydrographs. And so far, what we found is that wake boats, in particular, boats that have technology to make intentional wakes, as well as pontoon boats, which was to create the largest disturbance. But this is, again, ongoing, so we need more data.
Well, that's interesting. I think we're all very excited to hear what you're going to come up with, all of you, actually. And I'm sorry, we are just about out of time. Hannah, your questions haven't come in yet. I'm going to Still waiting. I'm sure they're going to come in. Hey, well, we'll get them to you. You guys have done fabulous research. I'm very impressed, really, and I'm really grateful for your time today. Again, I'll make sure we get more time next time. But in the meantime, we will forward you additional questions as they come in. And I would like to thank the audience too for sticking around, and we'll see you at the next session. Thank you.